Thanks very much. Thanks for coming along. It's a pleasure to be here and talk about some of the research we're doing at UTS and actually with a lot of colleagues around the place. So I'm going to talk about the possibility that blue-green algae could cause motor neuron disease. So blue-green algae is this stuff here that's floating on this river. In fact, it's not actually an algal. It's not an algae. It's actually a bacteria. It's called cyanobacteria, and it's one of the oldest organisms on our planet. So if we just start off by thinking a little bit about motor neuron disease, and we've touched on this today already, we've got about 10%, which we can say is familial. It's got some link with genes. And then we've got a 90% here that we don't really know. So Steve mentioned some of these genes. So the story is that your gene has got a pattern, and you make mistakes in these proteins. So you make mistakes in these proteins, and Steve also said they get a bit sticky, so they can fall out of solution, they can aggregate. But this is only 10%. We've got this other bunch here, which is called sporadic. So generally it's thought of as being multifactorial. So maybe we've got genes, but we've definitely got environmental factors. And someone asked a question about what sort of environmental factors. Well, with some colleagues out at Macquarie, we're writing a review on environmental factors in motor neuron disease. And we sliced it all up so everyone had a section to write. And I've read a lot of these papers, and the evidence is really, really very weak. So we've got things like diet, smoking, drinking, stress, fatigue, mobile phones, military service, welding, playing soccer. So this is quite important. So later on, I'm going to come back to this playing soccer in general sports, because there could be an interesting link, and it's got to do with this kind of stuff here. The problem is that we know a lot about this, but we don't know much about this. The reason being, with the genes, it's finite. You've only got so many genes. So with good technology, you can check every gene, you can work out every genetic mutation, eventually. Here, with the environmental, it's pretty much infinite. There's an infinite amount of environmental factors that could be causing disease, could be causing MND. It could even be you get exposed to things when you're in the womb. It could be you get exposed to things 10 years ago. So it's a very complicated area. However, let's say there was some environmental factor that by itself, it caused motor neuron disease. So I'm going to start off by telling you the story of Guam. Okay, so this is a story of an unusual disorder on the Pacific island of Guam. So it all started in 1521 when this Portuguese explorer called Ferdinand Magellan was sailing around the world. He came across this beautiful island in the South Pacific called Guam. He landed there, he met the natives, and then he sailed on. After they left the island was claimed by Spain, and then Spain ceded it to America. It's quite a strategic spot because it's south of Japan, it's north of Australia, and it's quite close to China. So the Americans liked it, and they started have a naval base there. So the Americans had a naval base here, and lots of American naval personnel and doctors were visiting this island. And they found out a strange thing. These are the local people, they're called the Chamorros. And the Chamorros had a really high instance of an illness which they called ALS-PDC. It was a very complex neurological illness. So it had elements of ALS, or motor neuron disease. It had elements of Parkinson's disease, and it had elements of dementia. It was 100-fold the instance anywhere in the world. So that was pretty interesting. But it wasn't actually confined to these guys here, the Chamorros, because there were Filipinos, Japanese, lots of other people there, and they also experienced this strange neurological disorder. So everyone thought, at last, we've got some really good clue about an environmental factor that could trigger neurodegenerative disease. And what else happened, which is interesting? When they started monitoring it, it declined over time. So wherever that factor was, maybe it wasn't there anymore. So something strange was happening. And they also found that the ALS and the Parkinson's dementia was a little bit separate, although some people experienced both types of disease. And it wasn't confined strictly to Guam. There were other islands nearby. There was West Papua in Indonesia, Groote Eylandt in Australia, and the Kii Peninsula in Japan, where they also found this increased incidence of this neurological disorder. There's actually only one report from Australia, and it was some paralytic disease that was experienced by Aboriginals in this part of Australia. And it came down to the possibility it had got to do with these palm trees. These are called cycads. And these three spots are part of the cycad belt. So these cycads, they grow in these three regions. And the indigenous people in the region actually use the cycad nuts. So if you chop one of these nuts open, it's got this white stuff in the middle. And the locals use this as flour to make tortillas. So there was a very clever woman went out to the island to try and study this disease. And she worked out that it probably had something to do with this cycad flour. And the locals actually knew it was toxic, and they washed it for a week. So they washed the flour for a week before they used it. If you don't wash it, it makes you sick. If you wash it for a week, it's absolutely fine. It makes a really good tortilla, seemingly. So what Marjorie Whiting did is she sent some of this stuff to a guy in England called Peter Nunn. And she said, Peter, I think in this flour there's something called BOAA, which had been implicated in another disease in Africa. So Peter Nunn was a young chemist there, and he had a look and he goes, sorry, Marjorie, there's no BOA in there. But he did find something else, something called BMAA, which is actually quite similar. It's called beta-methylamino L-alanine. So they found this molecule in this flour. So they thought, okay, let's give her a go. So what they did is, so here's Peter Nunn here. He's a friend of mine, actually. He's still semi-retired. So that was about, I think it was 38 years ago, this study was published by Peter Spencer and Peter Nunn. Peter Nunn made lots and lots of this BMAA, and then they gave it to macaque monkeys. So they actually put it in a tube, and they put it into the stomach, and they did this for about a month. And lo and behold, the monkeys developed MND-like symptoms. So everyone was thinking, great, we've found some environmental factor. It's going to give us a clue about one possibility for something that's going to cause MND. However, the problem was the amount of BMAA they used. You'd pretty much have to eat your body weight in flour every week to get that dose, right? So everyone thought, no, it's not possible. And then came along someone else, this guy Paul Cox. Paul Cox was a Ph.D. student of Oliver Sacks. Has anyone ever read any of these books, Awakenings? Okay, so Oliver Sacks, he's an author and he's a neurologist, and this was his Ph.D. student, and they went to Guam. And they went to Guam, and Paul is an ethnobotanist. So what he does is he goes to these indigenous cultures and he learns a lot about them. He learns, their habits, what they eat. So Paul went to Guam and he found out two really important things. The first one is the people in Guam loved eating flying foxes or fruit bats. They loved eating fruit bats, and he found out the fruit bats actually fed on the cycad seeds, and they bioconcentrated this BMAA stuff 10,000-fold. He also found out something interesting, that the BMAA was not made by the cycads, but it was made by cyanobacteria that lived in the roots. So this is the roots of one of these cycad palms. If you chop it open, it's green. And it's green because of that stuff that I showed you on the river in the first slide, cyanobacteria. So he uncovered two important things. BMAA is produced by cyanobacteria, it's eaten by the flying foxes, and it's bioconcentrated. So you go, well, why would people eat flying foxes? Well, the Chamorros in Guam loved flying foxes, right? And this is the recipe. You shampoo the bat, and then you boil it in gallons of coconut milk for 2 hours, turn it halfway, serve with rice. And one bat will serve 2 Chamorros. The interesting thing about this is we've later discovered that because when Paul went to talk to these people, they were saying, what is it about bats you love so much? And he goes, I don't know, it's like something you've never tasted before. And they also said that about the tortillas. And what actually happens is the BMAA gets converted into something in the body that gives you a bit of a buzz. So they were actually getting a little bit of a kick out of eating fruit bats and tortillas. And they actually wiped out the whole fruit bat population. At one time, they reckoned there was 100 fruit bats per square kilometer, and they wiped them out completely. Because originally, they just hunted them with nets, and then once they got guns, they just like shot them all and ate them. And they imported bats from nearby islands, but they didn't taste any good. They didn't taste any good because the nearby islands, the bats didn't actually feed on the cycad forests. So that was one interesting story about Guam. So what has it got to do with today? Because they wiped out the fruit bats, most people have recovered. What's the story? Well, the story is that BMAA is still in our environment. And a bunch of people in America, these guys are in Miami, they examined brains from people who had died from ALS or MND and Alzheimer's patients, and they found BMAA stuck in the brain. This is a little bit controversial because other groups have looked for BMAA in the brain and they couldn't find it. And one of the reasons is that how you process the sample is really important. So what we're trying to do at UTS with help from the government this year is set up our own lab where we can analyze samples for the presence of BMAA. And what we're going to do is we're going to send samples around the world to different labs and see if we can get an agreement on how you can detect BMAA. But 2 studies so far have found BMAA in brains of people who have died of various neurological disorders. Another really fascinating study was a neurologist who's in New Hampshire. I've never been to New Hampshire, but I'm told it's got lots and lots of lakes. And this guy called Elijah Stommel had lots of MND patients. And he had this bright idea of Google mapping his MND patients. So he put all these MND patients' location on a Google map, and I've seen this map, I've heard them talking. They were all round about these lakes. And the more interesting thing is, most of them lived downwind. So there was a prevailing wind direction. Most of them lived in the downwind side of these lakes. What it could mean is that you can actually inhale some of this coming off the cyanobacterial blooms, because they get a lot of blooms in these lakes. You get a lot of blooms like this around the place. You can see it in Centennial Park. You can see it everywhere. Thirdly, Paul Cox found that the American soldiers that fought in the Gulf War had a higher instance of ALS or MND. And he went out to Qatar a couple of years, 2009, and if you dig the crust off the sand, if you take the crust off the desert and you pour water on, it goes green. About 1/3 of the desert crust has got cyanobacteria living underneath it, just waiting for a bit of water. And just to come back to the soccer players thing, they actually use fertilizers made from cyanobacteria on grass sometimes. So there is a possibility that all that stuff about grass and things has got to do with cyanobacterial-derived fertilizers. I might come back to that a little bit later. Okay, so a couple of papers more recently. Bergman, she lives in Sweden, and she examined the Baltic Sea. And she sampled fish from every single level in the Baltic Sea and found that they actually bioaccumulate BMAA. So it's exactly the same as the fruit bats, and it's happening in the water. Another one is a guy called Larry Brand, who's from Miami. He goes around the country in a boat sampling seafood for various reasons, and he found that the levels of BMAA in pink shrimp and mussels in in Washington, were higher than the levels in fruit bats. So what I'm saying is the BMAA is still in our environment. The BMAA is still being concentrated. So if you taste any seafood and it tastes just a little bit special, you just never know. You just never know. So this is, I guess, is a bit of speculation, but what we want to do is we want to get a good lab set up so we can quantify BMAA really, really accurately in water, in seafood, and fish that we're eating. Okay, so is this a causal coincidence? Did BMAA actually cause the disease, or was it just there? Excuse me, was it there anyway? Is it possible that one molecule can cause like 3 distinct neurological disorders? So at this point, we have to do a little bit of science to understand how BMAA can do this. Okay, so protein synthesis is a process where you take information on your genes and your DNA and you make it into a functional protein. Okay, to do that, you've got these building blocks. So these are the 20 protein amino acids. And believe it or not, all our proteins are made from these 20 amino acids. So what's a protein? Well, proteins are things like insulin. It's a small protein or a peptide. Collagen. I got this from China, and it's a little bit dodgy. Collagen is a protein. So actually, you know, when you get old and you get wrinkly like me, that's because your collagen is starting to get a bit damaged. Your digestive enzymes, so you guys are going to hang around and have lunch. When you have lunch, you're going to get digestive enzymes in your stomach. These are all proteins. After water, most of your body is protein. And you make proteins from these 20 amino acids. So what they basically do is they form a chain. So these are all chains of these amino acids. This is actually the enzyme that degrades alcohol in your body, alcohol dehydrogenase. So you can see how it's quite a complicated process, and I'm just going to show you how it actually works. Okay, so here we have the mRNA. So this is the recipe for the protein. And what it contains is codons, and the codons match anticodons. So this codon here says, I want a methionine. So that's one of these 20 amino acids. So the tRNA carries the methionine, it locks in here. The next one says, I want a proline. The proline comes, locks in here. Suddenly, this chain is building up. So what doesn't happen is we don't make mistakes. When we're making proteins, we very rarely make mistakes. It's something like 1 in 1,000 or 1 in 10,000. It's really important you get this right, because if you get it wrong, you end up with some really dodgy proteins that misfold and can precipitate. The most important process is getting the right amino acid onto the tRNA. And this happens with an enzyme called the tRNA synthetase. So there we go. Okay, so this is amino acid. It's called valine. Okay, it pops along. It's got its own special little pocket in this enzyme. Along comes an energy source. It just opens up this pocket. Along comes the tRNA, uncharged, and suddenly a bond gets formed, and you've got your charged tRNA. So this is a really important process, because down at the bottom it says that's going to be a valine, and you want a valine connected. And the problem with this is that we don't make that many mistakes. That is exactly true. However, there's 20 amino acids here, but there's about 1,000 amino acids out in nature, and they're called non-protein amino acids. If you have a look here, they've got the same on this side, that's the acid group. They've got the same on this side, that's the amino group. So on top of these 20 amino acids, there's about 1,000 non-protein amino acids. Most of them are made by plants. And plants actually fight each other using variations on these amino acids. There's a type of grass called the fescue grass, and if you plant it, not many things grow near it. And one of the reasons why is it releases into the soil something that is not exactly tyrosine, but it's just like tyrosine with a slight modification, and that kills other plants. So these amino acids can actually be quite dangerous, the non-protein amino acids. Some plants actually use them to kill predators. So if the predators are feeding on the plant, it gets the non-protein amino acid instead of the protein amino acids, and it makes the wrong proteins. So fidelity in protein synthesis means we don't make mistakes in protein synthesis. And what is often forgotten is it relies on it being mostly protein amino acids in your body. So if you've got the 20 protein amino acids in the body, there's no problem. However, if you get invaded by non-protein amino acids that are capable of being charged onto the tRNA, you can make mistakes. So what we found in our research is that this BMAA molecule I've been talking about that's made by the cyanobacteria, it can mistakenly jump on the serine tRNA. So somewhere where it says, give me a serine, it'll get a BMAA. And what actually happens is, oh, sorry, I was going to say that this is published in a paper in 2013. I think it's about 6 months ago. And I checked the other day, this paper has had 10,000 hits. People are quite interested in this blue-green algae, MND. And this is the first time someone's offered a really plausible mechanism of how BMA could actually cause the disease. Okay, so this is what happens. Normally you've got your protein chain, it's got its characteristic shape. Instead of a serine, you pick up a BMA, what happens? You've got a different protein. So you've got a misfolded protein, and actually, if this happens for a long time, these proteins actually stick together and form aggregates. Almost every neurological disorder is characterized by aggregates in your neurons. Different proteins for different disorders. What happens if you put a lot of BMA in cells? They make these unusual proteins, and they start sticking together. And they stick together so much that they actually fluoresce in the cell. So if you stick the cells under a fluorescent microscope, you can actually see them fluorescing. And if you give the cells a lot of serine, you can prevent this fluorescence. So this is the research we're doing right now. What we're trying to answer is, where do these aggregates form in the cell? What proteins are involved? And can we prevent these aggregates from growing? Because one thing they do is they seed, and then they attract other proteins, and they get bigger and bigger, and the neurons stop working properly. Another aspect of our research has been done by Nick Cole. Nick Cole's out at Macquarie, and we're using zebrafish. Zebrafish are used quite a lot in neurological research, and what we're doing is we're injecting BMA into the zebrafish egg, the embryo. And the zebrafish, when it's developing, is sometimes using BMAA in place of serine. And you can see here in the normal fish, you get these fluorescent motor neurons. In the BMA-treated fish, you get shorter motor neurons and you get branching. So if you look at that on the graph, here's your normal fish, here's your BMA-treated fish, shorter motor neurons and branching. So we can use this fish to answer a few questions. We can say, is serine protective? And what we're just at this stage at the moment where we've looked at the normal fish with BMA and we've shown this type of effect. Now we can look at fish that have got genetic mutations. So we can actually get into that 90% where we can look at environmental factor, which would be BMA, plus genetic mutation. And we can try and work out a bit more about that 90% we know nothing about. So this BMA incorporation hypothesis is new, but it could explain a lot of things. It could explain why on Guam you got this ALS or MND plus Parkinson's plus dementia. Why would that happen? Well, what you're doing is you're introducing BMA into all these proteins. So in some cases, the proteins are going to misfold that are the ones that give you Parkinson's. Proteins that are going to misfold are the ones that give you ALS or dementia. Because all these diseases are characterized by an accumulation of misfolded proteins in your neurons. The latency of the disease, I didn't really mention this, but what happened to the people on Guam? Some went to live in America, and then 10 years later, they developed this ALS-PDC in America 10 years later. So what happened was something was initiated in their neurons that after 14 years on average gave a disease. So this is something that's always puzzled people about this latency of the disease in Guam. Finally, this is something else that is particularly interesting to me, because why is it nerve cells are affected? Because all the cells everywhere in your body are going to see BMA, they're going to use it in place of serine. So why would your nerve cells be affected? Well, the reason being is they live so long. If you've got cells that are turning over quickly, that are producing a lot of daughter cells, they'll actually distribute the aggregates among these daughter cells. So it keeps the aggregate load down. However, if you've got a neuron that you've got for life, it's going to keep accumulating these aggregates. Another cell that's long-lived are the retinal pigment epithelial cells in your eye. And I was really fascinated because I would predict then that the other cells that would be affected would be these epithelial cells. And when I was reading about this Guam stuff, I found out that there was some disease called linear retinal pigment epitheliopathy that was detected in the people at Guam before they developed the ALS or MND. So it was a predictor of later development of ALS. So obviously, for me, that was another indicator that maybe we were on the right track here. maybe these cells that are long-lived are really susceptible to the BMAA. So I'm just going to thank the team at the moment who are working on this. So the team in UTS have got Rachel Dunlop. She's a postdoctoral scientist. And if you have a look at, this is our website, Tellurian Toxins. And what we try and do is keep people up to date with what we're doing. And Rachel's been really good at this. Where does tellurian toxin come from? Well, it was a weird one because one of our papers was translated into another language and then it was translated back. And there was lots of really unusual words in there. And tellurian was one of these words. It actually means from the earth. So this is toxins from the earth. So the reason that we chose this for our website is that if you do a Google search on tellurian toxins, you've got us. There's no one else out there. So you can check this if you want to find out what we're doing. We've got Brendan Main, who's a Ph.D. student. We've got a couple of honours students. And really importantly in this work is our collaborators. So we've got Nicholas Cole, who's the zebrafish man at Macquarie. We've also got Professor Gilles Guillemin at Macquarie. And Paul Cox is one of our major collaborators. So Paul Cox, still actively involved in this research, as is Peter Nunn. So I've got time, yeah? Couple of minutes. I'm just going to finish on diet and MND. So this is only a couple of slides, but it's quite interesting. Okay, so this is a study that was done in 1982. They recruited more than 1 million men and women, and they got them to fill out a 44-item food frequency questionnaire. So basically they say, do you eat this? How often do you eat it? Do you eat this? And then they followed these people right up to today. And the idea was that what they were going to do is they were going to work out how many of these 1 million people died of cancer, and then they were going to look back and see what they were eating that the rest of the people weren't. It's a pretty clever study. Someone else had an idea. This is someone called Morzova. She had an idea like, why don't we go back and look at the same group and find out how many of these 1 million developed MND, and then go back and see what they were eating that everyone else wasn't. So I'm just going to share one thing with you from that study. So I'll tell you how it works. So does chocolate cause MND? Okay, so what you do is you've got a bunch of people who never eat chocolate, people who eat it about once a week, 2 to 3 times a week, or more than 4 times a week. You divide the number of MND cases by the total number, and you get an answer of 1. Okay, so people eat more than 1, 2, it stays 1, right? There's nothing. Chocolate is completely neutral. But you know the crazy thing? The crazy thing about the most positive result they got was chicken. Okay, so they found out if you never eat chicken, you've got a score of 1. If you eat chicken more than once, it goes down to 0.94, 2 to 3 times, 0.79, 4 times or more, 0.58. That was really highly significant. So chicken definitely gets a thumbs up. They couldn't really explain this. This is just crazy. The interesting thing is that out of all these foods they analyzed, they were really all close to 1, right? So it wasn't like there was a lot of noise in the information. However, if you look at meat, meat was sort of trending the same way, but it wasn't significant. Eggs, trending the same way, but it wasn't significant. So what do you think that's all about? Well, what I think it's about is the possibility that there's a number of these non-protein amino acids out there in our environment. And if you eat a lot of protein, you've got a lot of protein amino acids in your body. So you're actually, in a very, very long term, could be protected. This is a theory, so don't hold me to it, but it's what we're working on. So what we're working on is this hypothesis: the entry of non-protein amino acids into the human food chain and their incorporation into proteins might contribute significantly to neurodegenerative damage. Someone earlier on asked about MS. There's a really interesting guy who's 93, and I talked to him on Skype last year, and he's got this theory that a non-protein amino acid called azetidine is responsible for MS. And azetidine comes from sugar beet. So that's another interesting story, but I think I will leave with you the idea that non-protein amino acids can potentially cause the disease. It's a hypothesis. We're testing BMAA at the moment, and hopefully if you invite me back here again, I'll be able to tell you a bit more information on that. So thanks very much for your attention.
Thank you, Ken. That was very interesting. So questions from the floor, please. Got one just down here.
Repeat the question.
Sure.
That was very interesting. Question in 2 parts. What is serotonin? And secondly, how much exposure might you need to blue-green algae to be effective?
Yeah, two interesting questions. One is what is serine, and how much blue-green algae do you need to be exposed to? No one really knows how much you need to be exposed to. I think on Guam, for sure, these people were exposed to a lot of blue-green algae because they ate a lot of flying foxes. But if we find out, it's prevalent in seafood, they've actually found it in mushrooms in France. So anyone who's looking for it is basically finding it. So there is a possibility we could be getting exposed to way more than we realize, but we don't know at the moment. I think that serine can only be beneficial if it's taken at the same time as the BMAA. I think once you get these aggregates in your brain, I don't think that it's possible to break them apart using serine. I think the serine is protective earlier on, but if you think about it logically, once the aggregates are formed, it might not be possible to deaggregate them.
Okay, next question from the other side of the room. Oh, okay.
Yeah, thank you so much. I have a question regarding actual L-serine, which I've done a little research into, and there were studies done in America back in 2013, clinical trials, whether or not this exact amino acid was going to be of benefit at all. Have you got anything to add to that?
Yeah, I was sort of slightly involved in that because that was a trial that was set up by Paul Cox. And Paul is a great believer in, giving people a chance. And even though I said, I think it's too late, Paul. I think if these people have already got MND, I think it's too late to give them serine because the aggregates are there. He thought, well, serine, it's not toxic. Let's go ahead and have a go. To be honest, the study is still going on. I don't know the outcomes of the study. Paul also sent up another study with vervet monkeys in the West Indies, looking at the same thing, and that study is ongoing as well. So we're looking at that possibility. But I don't have any information on it. But yeah, serine's an amino acid. It's in your diet. It's non-toxic as far as anyone knows.
A written question up the back. Eileen, do you have one? Or? Microphone for Eileen. Oh, same question? Okay. All right. Another question down there. Yep.
5 years I came from England about 30 years ago.
I came from Scotland, by the way.
All right. Well, I can understand. All my life I've been a fisherman, and the last 3 years I've had MND. All the places I fish have blue-green algae.
Yeah.
And I also take home trout, which eat subaquatic animals. Should people be warned about blue-green algae in our lakes and rivers?
Yeah, no, okay, so someone just asked the question that they've been a fisherman, they've eaten a lot of fish, they've been exposed to a lot of blue-green algae. Should we warn people against this type of thing? Well, we're trying to convince the government to give us money to set up an analytical lab, and we've got chemists who are experts at UTS who are willing to do it, so we can actually check this out. Because at the moment, people are not really sure. It's hard to detect, and you need really quite sophisticated equipment. So hopefully in the future, we can actually, you'll see papers will be published, they'll say there's this amount of BMAA in these samples. What I'd really like to do is something like Centennial Park has got carp, and carp live for 60 years. So what we're going to try and do is we're going to try and sample the carp to see if we can detect BMAA in the carp, because they would have been swimming in waters potentially contaminated with BMAA. Was there a second part to that question? Oh yeah, I was going to say something else, that there's an island in Japan where people live longer than anywhere else in the world, and it's somewhere that Paul Cox visits quite regularly to study their habits. And what Paul has found is they do not eat seafood, which is crazy because it's an island. But the top 8 of the top 10 items in their diet is actually seaweed. And seaweed is actually one of the richest compounds in serine. If you look at serine levels in seaweed, they're really high. I don't think that BMA causes MND in everybody, but I think it's one causative factor.
Question?
I wanted you to go back to the eye.
Yeah.
Because my husband, who has MND, his optometrist noted degeneration of a nerve in the eye. I just wanted to name to that. And also the zebrafish, what symptoms do they display when you inject them with?
Yeah, okay, yeah, good questions. So the first question was the fact that BMA can cause changes in the eye. That hasn't really been documented. I've talked to a few optometrists and said, have you ever seen anything in people's eyes that, you might have to use a different type of approach to detect changes in the eye, but it is possible. But it's something that was really well documented on Guam, that they developed this really unusual disease. And it was sort of like linear. So it was like there was this like worm at the back of the eye. It was a weird one. Okay, so that was the first question is no one really knows. No one's really seen any changes. No one's really ever looked for any changes. Second question was, help me here. Oh, the zebrafish, yeah. Okay, so what we're doing in the zebrafish is the preliminary studies where we look at the development of the motor neuron, right? And then once we do that, we'll move on to the swimming experiments. So you can actually look and see if there's any change in the function of the fish. So what we've got is the high-speed camera that locks onto the fish, and you can actually monitor its swimming pattern to see the BMA-treated fish swim different from the normal fish. And you can also actually work out how long it takes the fish to swim against the current before they get exhausted, which is a measure of their function. So yes, we can do functional studies in fish. Thanks for the question.
Question? There's one behind you, Anne. Oh, sorry, Michael's.
I was just going to ask about to do with diet as well. From what I've heard, the incidence of MND and ALS is getting more and more than in the past. Whether things like processed food, chemically enhanced, whether there's ever been studies of BMAA in that type of food. Yeah.
I'm not sure about that, but what I do know is that this sort of global warming effect, we're getting more algal blooms. I know, especially like in Florida Bay, the amount of algal blooms they get there is absolutely phenomenal. So I think the way the climate is changing in the recent decades, for sure, there's the literature's out there that we're getting more algal blooms. So there is a possibility that we're further enriching our environment in BMA. How much of it carries through processed foods, I don't think anyone's looked at that, but it's a good question. Thanks.
Another question up there, Michael.
Yeah, hi. I've had tests for environmental toxins. What can I expect to get as feedback? Because it seems to be a fairly lengthy process in as far as getting feedback.
Yeah, I mean, sorry, the question was, gentlemen had tests for environmental toxins, and it seems to be a lengthy process, and what can I expect as feedback? I mean, I've read these papers on, heavy metals and MND, and it's really difficult because there's so many factors involved, and none of these studies have really been conclusive. I think Steve actually mentioned that none of these studies have ever been conclusive. It's just an area that's very hard to study. And also, even if they find heavy metals, and anyone that's exposed to heavy metals will have heavy metals in the body because your body doesn't get rid of them. But how you link that to a disease process is, then again, it's almost impossible. What I didn't actually say about the rugby players, because I talked about this at a neurological conference, and a lot of people came up to me and they had patients who were rugby players, and a lot of them were halfbacks. Does anyone support the Tigers? Was Scott Gale a halfback? He was? Played in halves. And I talked to this guy who was a neurologist of Van der Westhuysen, this South African rugby union halfback. and he was one of his patients. And we worked out that there's a possibility that halfbacks, they go close to the ground, and they lick their fingers, and they rub them on the ground to keep them rough. And they reckon, well, we had this idea that it's a possibility that when they use cyanobacterial fertilizers on the ground, you're ingesting it through your career. And the Italian soccer players, which were part of a couple of studies, same thing, they could have been inhaling it as they're running around. So there's a lot of possibilities like that. Another one is golfers. Quite a lot of golfers develop MND. And golfers sometimes, especially the old-fashioned golfers, if they were on the green and there was a bit of dirt in their ball, they'd just lick it off. So yeah, not recommended.
I think one question from this side of the room. Do we have any other written ones? No?
A friend of mine suggested to take chlorella supplement, but when I looked on the internet, it was connected to blue-green algae.
Okay, sorry, what supplement was that?
Chlorella.
Chlorella, yeah. I don't know about that, but I know that there's actually quite a lot of health foods that potentially could contain BMAA, potentially. Yeah. There's, I had a friend in America that wanted to check these, and she got a phone call from someone and said, "Don't you dare." Yes, this is serious, and I think that someone else in Canada actually either has published or is just about to publish a study showing that the contamination of certain health foods with BMAA, but I'm not sure if that's been published. But it's a big industry.
Okay, last question. Phil.
Thank you very much. Your presentation has been very interesting. But I have not got a question, but a comment. Fisherman got the guy in front of me, and I remember in my early 20s was fishing at the fish river dam at Avon, and there was a drought on, and the water was stagnant. I had a little algae in it, and I was by five in the water, and I got an eye infection. That's my comment.
Okay, yeah. So the gentleman commented on when he was younger, he used to swim in water that was heavily contaminated with blue-green algae, and he got an eye infection, so he would say potentially been exposed to that. I had an email recently from a guy from Germany, a teacher, and he was interested in this whole story. And I said to him, just out of curiosity, have you ever lived near a lake? And he goes, oh, yeah. We used to swim in a lake when they had these algal blooms, and we'd come up covered in green, and we'd call ourselves the Green Man. So yeah, there's a lot of algal blooms around, but I'm not actually 100% sure. Most of these cyanobacteria can make BMAA, but unless we test, we're never really sure that any algal bloom is making BMAA at that time. So yeah, thanks for the question.
I'm just going to take one more. This lady just down here, because I've missed her twice now. I can't say no.
My daughter never played soccer, but she's played softball for many, many years. Now, would there be the same problems on the fields with those playing?
So the question is, if you play softball rather than soccer, would there be the same problems in the fields? Well, I would say there would be the same problems if you're playing on grass, and if they use a fertilizer that's based on cyanobacteria. What is not very well known is in China, sometimes they dig the cyanobacterial mats into the rice fields as fertilizers. So yeah, using cyanobacteria as fertilizer it's not totally uncommon. But yeah, thank you.
Okay, so once again, thank you, Ken, and I'm sorry we couldn't get to everyone's questions, but I think we've got some very good answers. So thank you very much, a very interesting topic, a very token.
Thank you. Thanks a lot.